that was good, John. You were able to define for me what GLaDOS is, so I can skip that slide. So this is a bit about biologists, sort of voyeuristic biologists trying to discover the secret lives of fish. And some of those voyeurs are Tom Binder, Todd Hayden, Daryl Hondorp, and Chris Vandergoot. And Daryl and Chris are in the room, so co-conspirators. One of the things characteristic of GLaDOS is that we have lots of agencies that are involved in collaborators, in a full sense too, not just sort of hand-waving, but true collaborators. So that's a great thing. Let's see, this should work. Point at the computer. Thank you, Michael. GLaDOS, the Greek goddess of acoustic telemetry. Okay, so what's acoustic telemetry? Probably lots of you already know this, but I'll run through this just a little bit. They got tags, and we put the tags in fish, and they emit a sound that's unique to each individual fish. These, as you can see, are some fairly small ones, and even smaller you can get down to V7s, and then changing your frequencies go to V5s that are even smaller yet. But for like our walleyes and sturgeon, we often put the larger V16s in, and if you program them right, you can get 10 years of life out of them, so quite a long time. They're surgically implanted, as Daryl's doing there, into the belly of a sturgeon, and then these fish are released into the water, almost always immediately released. So then the fish is swimming around in the water, and we have hydrophones, or receivers out there that are listening 24/7. And when the fish swims by, the acoustic signal goes out, that's recorded, and it's combined with a date and timestamp. So we typically put these receivers, these are these black cylindrical things, in lines, like perpendicularly out from the shoreline or across a bay. Alternatively, we can put them in arrays. And if the receivers are time synchronized, exactly, you can look at the difference in the receiving of a particular signal that's emitted, the time differentials, do some fancy math and come up with a 2D position. So kind of putting this together, we'd have a receiver on the bottom and a tagged fish swimming by. The receiver's listening sort of in a cone. A fish swims by and emits the signal. Date and timestamps recorded, and then we've got to get that receiver back and download the data off of it. So we hit a button on an acoustic release, and the receiver comes to the surface. And this is the way in which we would do it in deep water. Shallower water, we might have a buoy on them or a snag line or something like that to recover the receiver. Design considerations in projects like this, well, like any piece of science, you need some sort of research question, and ideally, you can convert that research question into some type of hypothesis and then consider what would be the data that I'd like to see to support that hypothesis, and we might call that a prediction. We've done this sort of thing for all of our core projects multiple times over. Other sorts of considerations obviously include the tags, the type of tags that you put in, and how you program them. How frequently are they going to transmit? Now, I've already mentioned receivers. There are some different types of receivers, but also then the number, the type, where they're deployed, how you're going to get them back. Are you going to be out there just during the season? You're only interested in the reproductive period, or do you want them out there all year round? Obviously, the fish that you capture is another important size, number. You're going to catch them at spawning time or from the fishery and tag them. And we're looking at a new project on Lake Erie, where we're going to be tagging in the fishery during the middle of the summer and then seeing how they disperse to streams during spawning season. So it's kind of a reverse then from what we've been doing in the past. So today I want to talk about GLaDOS, the organization, what its current status is today, future challenges, and then most interestingly, at least to me, is to actually look at some data that's coming out of some of these projects. So GLaDOS started with monies that came through the Great Lakes Restoration Act initiative, monies that came to the Great Lakes Fishery Commission, and over a 5-year period we have received a lot of money, $6.4 million. And when we first got this going in 2010, we established 3 large projects on sea lamprey, walleyes, and lake trout, and then a 4th in 2011 with some additional funds as well as GLRI money from the Great Lakes Fishery Trust, and that one is focused on sturgeon. So in that initial mix of 4, I think we had a pretty good group of projects. We had some that were that fine scale, using those arrays and getting 2D positions, and that included the sea lamprey project, lake trout, and walleye. But we also had large spatial scale type of projects where we were using mainly these receiver lines, spatially extensive from northern Lake Huron out into Lake Erie. And that would include then the sea lamprey, walleye, and lake sturgeon projects. In terms of relevance, these addressed native fish restoration, invasive species control, and then the walleye project certainly could provide some information that may inform things around quota management for the walleye fishery in Lake Erie. Okay, so it's 2010. We're trying to get really largest projects that I've ever been involved in up and running. Immediate challenges we had these 4 projects. We knew with the new technology other projects were going to come into the basin. How are we going to interact with those projects? We have receivers that are out there, each for these individual projects, but if I've got my tags might be going by someone else's receivers, and I'd love to get those detections back. So there needed to be some sharing. We need to know where the receivers were being placed and what their operational schedules, and then be able to share tag detection data. We knew the public was going to interact with some of our fish, like lake trout and walleyes. We'd love to get the tags back to know where that fish was caught. How are we going to make that happen? Thus, really communication became a central focus immediately. Not only communication among the leaders of the projects, but also communication with the public. And if you think about these receivers out there listening constantly, and you've got a tag and a fish is swimming by and it kind of hangs out for a day or two and it's transmitting every 2 minutes, you get a lot of data. And we got tons and tons of data. So the strategy we used was to learn from other strategy. We didn't really have a lot of embedded people in the basin that knew a lot about acoustic telemetry, and I didn't see any need that we needed to reinvent the wheel, but we could go out to the coast particularly and learn. We lacked expertise, and so we were able to go out to the USGS lab in Columbia River in Washington and capture Chris Holbrook and convince him to come and work at Hammond Bay. We got Steve Cook out of Carleton University. He'd been working in British Columbia lots on the Fraser River, but we got him interested in Great Lakes topics, and recently got Aaron Fisk involved from the University of Windsor. He worked lots in the Arctic. So importing some of that expertise is one of the strategies. I went and talked as well to out the Post project. It's a tracking project from Washington to Alaska on the continental shelf with receivers. So I talked to David Welch and then the 2 directors. First, Michael Stokesbury. He actually talked at an annual meeting for the commissioners a few years ago. Directors of the Ocean Tracking Network. And that one is global. I mean, we're talking South Africa, Australia, South America, obviously North America. Now Fred Wariski is director of that and been in lots of contact with them. We essentially adopted an OTN approach to trying to set up GLOTals. So obviously we're not global, but still it's the Laurentian Great Lakes, pretty good sized body of water. And that's the genesis of GLOTals. So what's the focus of GLOTals? Well, we did run the mission statement. GLOTals is a network of researchers that develop partnerships so they can share detection data. It's more than that, but that's our succinct sort of statement. We had those 4 projects, but we already knew that GLOTals was not going to be those 4 projects. We wanted to include everybody that was using this type of technology in the Great Lakes, so it was to be broader, to try to incorporate everybody in. And I've been to some meetings where projects like on the South Atlantic coast, and they didn't have any organization like that, and it's just a horrible mess of trying to communicate and share tag data with their saltwater fishes. So again, trying to learn from others. We did this through having annual meetings. We began in 2010. We've had one every year since. We also use a website as a tool for communications. And that was hosted on the Great Lakes Observing System website. And in fact, Gloss funded our website construction, and we're currently preparing a proposal for a revamping of that website, which will make it even better. Website's got lots of information. Go there if you want. But each of the projects has like a one-page project description. So the current status, we have 16 projects that are sort of in this umbrella of GLOTOS. 8 of those are outside projects. They're projects unrelated to the Fishery Commission or GLRI funds. So fully 50% now of GLOTOS are these outside projects. Within the inside projects, if you will, we have 3 sea lamprey, 2 lake trout, 2 walleye, and 1 lake sturgeon project. We've had 30 species tagged. More than 4,000 fish have been released. In terms of receiver deployments, we've had 3,000 receiver deployments at 1,200 plus receiver stations. So, and this is just a map of the receivers that are currently in the GLOTOS network across the basin. And then in terms, I said data, so we have 135 million total tag detections in the database. Now, on these receivers and with lines, we typically put a tag that's beeping occasionally. It's anchored out there, and that tells us something about the probability of tag detection. So if we take the sync tags out of that database, then we've got 69 million that are associated or matched to fish. So with that sort of infrastructure in place, we're getting new projects and things sort of being proposed, and we've got receivers in the inventory, and people want tags or receivers, and would you loan us some, and that sort of thing comes up to me all the time. So one of the things we've had to kind of beat back is this idea that, well, we'll just tag some fish and see where it goes. I say, well, that's interesting. Maybe not. Tell me what your research question is, and what sort of process are you thinking is driving the behavior of this animal? Try to move it in that direction. And sort of a corollary to that is that every fish released and every receiver deployed must have a research objective associated with them. And I would prefer to see a question, a hypothesis, a prediction, and a research objective. So we've held pretty tight on that as best we can. Our other agencies supporting GLOTOS, and several of you in the room have helped us in this, and I would say absolutely unbelievable in-kind support. Things like receiver tending, capturing of fish, even purchasing of receivers. It's quite remarkable. A tremendous amount of help, like Michigan DNR and Ohio DNR. Ontario Ministry did some original outside purchasing of receiver source. So pretty amazing, I think. Well, what about the science side? Okay, so publishing papers, that's the currency of science. Are we doing any of that? And this is what we've got so far. We have 11 papers, granted 10 and 11 are submitted, but I have high hopes that they will be published. And maybe if you think about it, we're in this just in the 5th year now. You need to have at least 3 or 4 years if you're going to say you have behavior that's repeatable and happening, and you're trying to associate it with some environmental variables or that type of thing. So it's maybe not surprising that then out of that 11, that 4 of them are actually about methods. We're trying to get a bunch of the receiver probability type papers, tag detection probability papers done and out, so that then we can just cite them when we get down to the actual fish behavior papers. Future challenges. 69 million tag detections. That's a challenge. It's a blessing and a curse. And the major problem here is trying to filter your data set down to some point so that you can actually do the analysis to answer the question you're after. And we were working with Stephen Riley on a site fidelity question with lake trout, and I thought, well, this is a month. Like 5 months later. We're still filtering data. By the time we got the filter finally figured out, it was just turn the crank and write the paper. I mean, we had the research question so well defined. It was quite easy, but it was all about trying to get down to that final data set. One of the things is access to the data and running this. It's quite a task. So we've just recently hired a data researcher or manager, and that's through a GLOS, USGS, GLFC, Michigan State partnership. So we're going to be able to even address this issue better in the future. Future challenge, it relates to all those data, and that's getting from data analysis to paper writing and publishing. We've developed detailed plans for each of those core projects, and this will just be an example. This is one that Daryl's working on right now. Maybe it'll get submitted by April 1st, or maybe not, but he's got a draft up already. But this is, again, sort of a methods paper about post-surgery effects on sturgeon. But we have this for about 50 or 60 papers out of those 4 projects, this type of level of planning. And what about the continued life of GLOS? That'd be another challenge. We have walleye and sturgeon tags out there that are going to be beeping out to 2024. Should we have receivers in the water listening? If we're going to do that, we're going to probably need to find some more permanent funding. Approximately, when you look at trucks and boats and receiver tending and tag replacements and receivers that are lost and all that sort of stuff, and people, technicians, so we have salary monies on there. There's about $500,000 in just program costs. And then if we want to do new projects, there'd be some additional funds. It does indeed feed on lots of money. Maybe that's the sinkhole that John referenced. But I think the good news is we're seeing some funds come in from sources other than the Great Lakes Restoration Initiative, Almanar and New York State, Michigan, Ohio Sea Grant, and so somewhat ancillarily, we got $1 million from the National Science Foundation, and that's to build small fish robots so that we can get past having stationary receivers and have active tracking in the Great Lakes. So we'll see where that goes. We might get $800,000 from GLRI in 2015, but I think that's the end. I would be very surprised if we get additional funds beyond that. But maybe we've got some momentum going, and maybe the program is starting to sell itself. Did I skip one? No. Okay. Last challenge, I think, and that's related to this money issue too. We need to make ourselves valuable. Are we valuable, or should we have done any of these projects, or was it a huge waste of money? Is there any effect from these projects on management? Did we improve sea lamprey control? Have we sped any sort of restoration for lake trout or lake sturgeon? Did we have an effect on quota management of walleye? So in summary, GLOS is growing. We've got new projects and new investigators coming in. We've started to publish, and these are critical products to produce, certainly. We've got broad support. Many of you in the room have been helpful, but we have federal, provincial, state, tribal, university folks involved. GLOS results, trying to make that relevance to the fishery agencies, have been presented at the lake committees each year. Chris Vandergroot presented here Monday morning. Stephen Riley's going to talk on tomorrow at Lake Superior or Lake Huron on lake trout in Drummond Island. So, plus I'm giving a talk. So there you go. We're trying to connect again and get the information out. So maybe we have some momentum. I do appreciate, like New York State's a new one. I mean, coming up, these are tough times to offer up money to make some of their projects go, and I appreciate that. Offering scarce funds of their own to pursue some of those questions. So maybe that funding is growing, and maybe there's some momentum. Now, the cool stuff, right? What are some results, some project tidbits? If you didn't get to see Chris's talk or see Stephen's talk tomorrow, I want to talk about lake sturgeon, lake trout, and walleye. So first, lake sturgeon. I think this one really shows the level of coordination, collaboration amongst agencies. Just a huge amount of help from everybody, especially in trying to catch the fish and to tag these fish and tend receivers and everything else that's gone into it. 268 sturgeon have been tagged, and these were with V16, so that larger tag, and they have a 10-year time life, or 10-year life, surgical implantation and then release. 3 areas were fished during the spawning season. So Detroit River, the lower St. Clair River, and then up at the upper St. Clair River. And these are suspected spawning sites. So 267 of the 268 individuals have been detected in the receiver network of 95 units in that Huron-Erie corridor area. 2.7 million detections since 2011, and a median detection per individual of 2,600 with that range. You're on. So the black are the Detroit River fish, red, lower St. Clair, blues, upper St. Clair. I thought the Detroit River fish would go out to Lake Erie. We've had a couple detections out there, but pretty much the Detroit River fish all stay in the Detroit River. It seems to be a river resident strategy. Somewhat the same with the red, the lower St. Clair River, yet over half of the blue, which is the upper St. Clair River fish, go out into Lake Huron. So we have some different life strategies in sturgeon that's going on. We see some, this is going into spawning season, but when you get into the winter, you'll see that it looks like also Lake St. Clair may be being used as a wintering habitat for sturgeon. Start to think about what this very brief few second clip, how much data is being displayed over time. So that's the data. Okay. Now we're in the winter and you can see the balling up there in Lake St. Clair. Okay, lake trout, Lake Huron. Again, lots of collaborators. Cora's been involved in this. Again, lots of help. This is a project where we're doing arrays. So it's what's on your right side here. So it's 2D positioning. Receivers are only placed out seasonally during spawning season. Objective was to compare wild and versus hatchery origin lake trout. So the site is the Drummond Island Refuge up there in orange, and where the star is where Horseshoe Reef is, and the dots are where our receivers were placed. This is the largest positional array in the world. It covers 27 square kilometers. We instrumented it to monitor environmental variation of temperature, waves, and wind. We had 393 tagged trout, and that was split evenly between wild hatchery male and female. So one of those questions was site fidelity. Do these lake trout keep coming back, and is there a difference between wild and hatchery fish? So from a restoration standpoint, I see some of this as a fairly good news. And the gray is just a hatchery female as an example, and the white is a wild female. Essentially, the hatchery fish returned to the site at about 80%. So I thought that was quite a lot. So once they find a spawning site, they actually come back. Wild and males and females both come back at over 90%. So they're show a lot of site fidelity. These dots represent average positions in 10-minute bins. Okay, so start thinking again the amount of data just for 2 fish that we have. And this is over 2 years. So you saw 2013 and 2014. Interestingly, both fish came in from the west both years. I don't know if it'd be interesting to look if there's a continued pattern like that. An example of how sharing receiver tag detection data helps us, the Lake Trout Project is all about Drummond Island. But these are the receiver detections represented up and down the coastline of Lake Huron from Todd Hayden's walleye project. And we had 40 fish over in Thunder Bay that were tagged as well. And we get some idea of how these trout distribute from their spawning grounds. And you can see that for the most part, the Drummond Island fish stay up north. Very little interaction with the Thunder Bay fish that tend to stay right around Thunder Bay. So that might have management implications too, in that what you do in one place in the lake may not affect that same group of fish. And last, walleyes in Lake Huron, and you know that project's a lot bigger, involves the Huron-Erie corridor and lots in Lake Erie, and Chris has already talked about that. But so where do walleyes disperse after spawning? Tagged in the Tittabawassee River and Eau Gray River with V16s, about 260 in the Tittabawassee and about 50 in the Eau Gray. What we found was 3 life strategies within the same spawning population, and I think that's pretty interesting. So there's a sort of a left-hand turning northward walleye, southward right-hand turning walleye, and then walleyes that just stay in Saginaw Bay. And then they all return about October, November, and they overwinter in Saginaw Bay. Okay. So you'll see them move out from the Tittabawassee, and they, as soon as they're done spawning, I mean, they shoot all the way up to the bridge, and they go down into southern Lake Huron with a bunch staying in Saginaw Bay. Now, they all come back about early November, over winter, and then when you hit March, boom, up the river and out, and they just, as soon as they spawn, they blast out and go, and then a bunch stay in Saginaw Bay. And that pattern, what's interesting is if you're a left-hand turning fish, you're always a left-hand turning fish. You don't switch in these strategies. If you're a Saginaw Bay stayer, you're a Saginaw Bay stayer. So there's a pattern at a population level, but also a pattern at an individual level. Okay, some final reflections, and that kind of leads us to this, is which behavior to describe. So with the biologist, and Michael was ragging once upon a time up at Hammond Bay about this problem, it's the look at this, what this one did sort of syndrome. You know, like, whoa, this guy went 1,000 miles, you know. Well, it's fun to look at the odd behavior, but it doesn't describe the population. So it's sort of the tail of distribution syndrome. I alluded to this already, is you got both population behavior, but also individual behavior, and the two can corroborate what you're seeing. But we see this now coming through this individual repeated behavior, and then there's sort of the average typical population behavior. From a programmatic thing, I think you got to ask, why do we have widespread collaboration? I'm sorry, but scientists aren't typically getting along with each other normally. They sometimes fight about things. They don't want to share data. We have widespread sharing of detection data. The reason this works is a super positive feedback loop where if I share data off my receivers, I get data back on my fish. So it's a win-win situation. Certainly the mountains of data, every one of the project leaders have said, I am overwhelmed. What are we going to do? And it gets to a point where it can be a problem to even get down and do an analysis and write a paper. And that leads us then, I think, to the oddest thing that you don't see among scientists. It's sort of like, take my data, please. Okay, so they're all looking for someone to do analysis and write papers. So, thanks. I'd be happy to answer any questions.